Okay, I just want to briefly say a little bit about the human genome. I think that most of you are aware since about 2000 with the sequencing of the human genome that the DNA sequences of everyone, 99.9% of them are identical across all humans. That leaves that half of 1% of 3 billion means that there are about 15 million bases that are different. We also now know of that 15 million that only really 3 million account for almost all of your differences, whether it's in the color of your hair, color of your eye, also in terms of whether you're susceptible to disease, etc. So really it's the 3 million single nucleotide polymorphisms that we refer to as SNPs that will really determine and what we're all about in trying to understand what they do. So, sorry, I'll quickly recap that those 3 million SNPs account for most of the things that regulate at the genetic level what you do, and also in terms of human disease. Now, you might, for interest's sake, know how and why we have these variations. And I think it's important to recognize as we go forward in the future, because I do think that sequencing your genome will certainly be a part of reality in the next 10 or 15 years. We know that the human race broke away from primates about 6 million years ago. And so over that 6 million period, the human evolved into 12 different species. And the modern species, of course, of Homo sapiens is regarded by us today as the modern human. So it has had 6 million years to evolve through those various species, and it's hard to imagine, but you have to think in terms of millions of years. And the part of this, in fact, where those errors come from, your DNA breaks down every few days, and you get new DNA, and when you get the new DNA, it is very carefully copied so that it only makes one error for every billion bases. But keep in mind, you're talking about billions of bases in little over 100 trillion cells that make up the body. So even at one mistake per billion, over the years, millions of years, these mistakes begin to add up. And all 94% of those errors are again single nucleotide polymorphisms, and that's why all of us as humans have 3 million polymorphisms that make up for most of those differences. They are derived from errors in your DNA. So if those mutations occur in the sperm or in the egg, they are passed on to the next generation and so then become mutations. So despite this low error rate of 1 error per billion bases, this leads to a mutation rate of about 1.2 times 10 to the minus 8, resulting in 40 new mutations per generation. In a human population of 7 billion today, that means we now at the moment in this generation have about 300 billion mutations, almost all of which are present as single nucleotide polymorphisms. With time, of course, these mutations, if they are favorable to life, they will become more common and stay around. If they're not, they're either gotten rid of or they remain very rare, and many of them are benign and neither beneficial nor in any way deleterious. All of you are aware that it's been known for about 50 years that genetic susceptibility for coronary artery disease is about 50% versus environmental factors. It is postulated by several investigators, and I'm sure some of you are aware of people like Yusuf and others, who have claimed that coronary artery disease is completely preventable and that could indeed be the last century for heart disease. Now, most of it is based on the fact that in clinical trials where we modify risk factors, we consistently see 30 to 40% reduction in cardiac events and in death, showing that in fact, if you could prevent the risk factors, it is a highly preventable disease. Now, I've told you that about 50% of that risk is due to genetic risk factors. And so if you want to comprehensively prevent coronary artery disease, it's clear that you must also treat those genetic risk factors in addition to the ones that we know, such as cholesterol, blood pressure, or diabetes. Up until a few years ago, we did not know what those genetic risk factors were. In 2005, the microarray was made available and gave us the technology to pursue genes related to coronary artery disease and myocardial infarction. I think many of you are aware of the fact that simultaneously the group in Ottawa and the group in Iceland discovered the first genetic risk factor, 9p21. As you can see here, it's a very common genetic risk factor. It's present in 75% of the world's population. It's a risk factor in all groups except for African and African Americans. And so that means that there are several billion people today who carry 9p21. If you have 2 copies, one from each of your parents, it increases your relative risk for coronary artery disease by 50%. If you have 1 copy, by 25%. I'm sorry that this is getting to be such a difficult story. The other is in premature coronary artery disease, 9p21 carries a risk of about 2-fold. And as you can see here, that from 2007, when this was initially published, it was confirmed around the world over the next 2 years in well over 64,000 cases and controls showing that 9p21 was a risk factor for coronary artery disease. And as I indicated, all of the various ethnic groups carry 9p21 as a risk factor, except African and African Americans. Following this episode, many of the groups, we decided to work together because we recognized it would require large sample sizes of hundreds of thousands, and it would also be very expensive to do DNA mapping and sequencing on when it comes to 100,000 individuals. So we formed a group with all of the various universities, both in England, Germany, in the US, and also with 2 institutions in Canada. This was the largest collaboration in cardiology ever known. As you can see here, we had a total population of 210,000, 100,000 controls, 100,000 with proven coronary artery disease, and it meant that each one of them now would undergo about 11 million markers that would be analyzed on each individual DNA of over 200,000 individuals. So to be short, we identified 50 genetic risk factors. All of them have now been confirmed in independent populations in all of the various ethnic groups, and the only one that is unique is 9p21 to the African American, who doesn't have it as a risk factor, but all the others have been shown in the various ethnic groups. Now, I'll summarize briefly each one of those genetic risk factors. averages only about a relative increase of 17% your risk for heart disease. They're very common. 50 of them occur in more than half of the population, and about a third of them occur in 75% of the population. This is, of course, what we expected because coronary artery disease is a common disease. You would expect that the genes that are associated with it would be very common. What was surprising to us, and perhaps the most important thing I think for everyone to remember from this conference, is that 35% of those genes are associated with mechanisms that currently we do not know. They do not operate through cholesterol or blood pressure or diabetes. So this implies that many of the mechanisms responsible for atherosclerosis and myocardial infarction have yet to be identified, and so this certainly is an important point in terms of the work we have to do to understand atherosclerosis. 15 of the genes do act through known mechanisms such as cholesterol, and as you can see here that one of the ones was PCSK9, that I shall come back to later. Only one of the genetic variants was directly associated with myocardial infarction, and that's the blood group A and B, and that is what causes thrombosis. I just, in the little time I have, what have we learned from indeed identifying these 50 genetic variants? First of all, as you can see here, it's not one genetic variant, it's how many of those you have. When you look at the 15,000 people that we plotted with the genetic variants, we observed that up to an average person having 17 of them did not separate cases from controls. But once you get past 17, you start to see people who have 20 or more than 20 and 30 of these genetic risk factors, then they become more relatively increased risk of having coronary artery disease. So the first observation is it's the number of genetic variants you have rather than any particular one. Now, we also did the same analysis looking for genetic risk factors that regulate plasma, LDL, HDL, and triglycerides. And we combined about 200,000 that we mapped, and we were able to, as you know, this was published just recently in Nature Genetics, that 157 genetic risk factors regulate your LDL, HDL, and triglycerides. It's been well recognized, certainly for a long time, that the cholesterol is 80% synthesized in your body. And that synthesis, of course, is regulated by a group of genes. And so you're not surprised to know that in the case of LDL and HDL, that 80% of the plasma levels depends on your genes. And these genes, as you can see, so we then looked at some very interesting features about them. And we found that consistently the genes that regulate triglyceride were associated with coronary artery disease. As for some time now it's been questioned whether plasma triglyceride is a risk factor for heart disease. But from the genetic point of view, we consistently observe that plasma triglyceride is a risk factor, and this has been confirmed in several other studies. Sorry, it's not always moving. Okay. It's also clearly established that HDL cholesterol, for all intents and purposes, protects us from heart disease and from myocardial infarction. This has certainly been taught in all textbooks for the past 30 or 40 years. In fact, since 1966, when Goffman proposed that HDL was protected. Now, one of the things about HDL is that all of the studies showing that HDL cholesterol is protective have a confounding effect, as you are aware of, because when you look at the evidence, it shows that statins will increase HDL, but of course they always decrease LDL. The same for niacin, the same for exercise, the same for alcohol. All of them increase HDL, but they also decrease LDL. So it's never been clear, is HDL per se a protector? So what we did to look at this factor, we picked a particular gene that is associated with increased HDL, and they've been exposed to that increase all their life. And we also picked for control some genes that are associated with an increased LDL, which we know is bad for you and predisposes to heart disease. So I won't go through all the details. Except these studies clearly indicated that a lifelong exposure to increased levels of HDL with a sample size of over 150,000 showed no protection from heart disease, from myocardial infarction, or from atherosclerosis. And these studies, as you know, in the last year have been confirmed by a couple of other studies. And of course, prior to this, there were several clinical trials trying to see and assess the effect of increased HDL cholesterol. And of course, none of them have shown benefit. So for the moment, it's clear from our genetic studies that HDL cholesterol per se is not protected. Now, I would urge you to say that we are only looking at HDL cholesterol. The claims that Gothman made in 1966 was based on HDL mass. But because HDL mass and cholesterol seem to go hand in hand, since that time, all we ever measure is HDL cholesterol. So it may well be there is something in the mass of HDL, but clearly all the genetic studies show us no association between HDL and protection from myocardial infarction or atherosclerosis. The other thing from these studies, clearly one of the things you want to do is to develop new drugs. And one of the mutations is PCSK9. PCSK9 has a very important role in the plasma in that it removes the LDL receptor and tends to increase your HDL, your LDL rather. So recognizing from this, studies show that if you lost the function of PCSK9, your LDL would be removed faster and you have lower levels of LDL cholesterol. And as you know, this is showing you one of the families that loss of function in PCSK9 was associated with 28% reduction in plasma LDL, but because they've been exposed to all their life, even with that small reduction, that was associated with 88% decrease in risk of heart disease. I think most of you are aware there are at least now 3 companies who have developed antibodies to PCSK9, and they're all in phase 3 clinical trials. All trials are showing 40 to 50% further reduction on top of the statin decrease in LDL. And so far, the adverse effects seem very acceptable. It looks like that it certainly will go forward in the FDA. So I think that what we have to look at in the future, one, is to find out what all of these fragments are doing, these DNA genetic fragments that we do not know their mechanism. And 2, certainly once we find the pathway, hopefully we'll develop new drugs such as the PCSK9 inhibition. But keep in mind that I think the age of knowing your DNA sequence is very much a part of life. At the moment, over 30,000 people in the United States have had their genome sequenced. It's estimated by 17 that over 15 million people will have had their genome sequenced. And we as physicians and cardiologists will have to know something about what are we going to do about it. I'm not sure I know at the moment, but the point to be made is that the cost of sequencing DNA has decreased 1 million-fold. And with all of these people getting their DNA sequencing, you have to recognize the fact that there is a big gap in terms of what this information stands for and being able to understand it. And while the rate of DNA sequencing is increasing and is fast, our ability to analyze and interpret the data has only increased 16-fold. It certainly will be a big issue. I'd like to give my congratulations and appreciation to my colleagues in the cardiogram setup and all of those in Ottawa who did a lot of the work. Thank you very much.